All right, let's dive in. Today, we're going to talk about a brilliantly clever solution to a really fundamental challenge in science, and that is, how do you sort things that are so incredibly tiny you can't even see them? It's a problem that shows up everywhere, from medicine to making new materials, and the answer, you'll see, is surprisingly elegant.
So just think about that for a second. How do you sort a mixture of things you can't even see? It's kind of like trying to separate specific grains of sand from a cloud of dust, but on a molecular scale. This is something scientists have to do every single day. It sounds almost impossible, but stick with me. And look, this isn't just some abstract puzzle they're trying to solve for fun. Being able to separate molecules is absolutely crucial for just about everything. We're talking about purifying life-saving medicines, studying the proteins that make our bodies work, analyzing the plastics we use every day. Getting a pure, clean sample is often the very first and most critical step to any big discovery. At its heart, the solution is something we can all understand. It's basically a filter, a sieve. But, this isn't for draining your pasta. This is an incredibly sophisticated molecular sieve, and it's designed to sort molecules based on one really simple property: their size. The official name for this whole process is size exclusion chromatography, or SEC for short. Now, that sounds pretty technical, I know, but let's just break it down. Chromatography is just a fancy science word for techniques that separate stuff. And size exclusion, well, that tells you exactly what it does. It separates things by excluding them based on their size. Simple as that.
Now, here's where things get really interesting, and honestly, a little backward from what you'd expect. You'd think the small stuff would just zip through a filter first, right? But with SEC, the exact opposite happens. The big molecules are in a hurry. They're too big to fit into all the tiny pores in the filter, so they just take the express lane, the shortcut around everything. And they exit first. The little molecules, on the other hand, they take the scenic route. They wander in and out of all those little pores, which really slows them down. So the biggest molecules come out first, and the smallest come out last. Wild, right? So let's walk through this molecular race step by step. First, you inject your jumbled mixture of molecules into a column that's packed with this special porous gel. Then, a liquid carrier, scientists call it the mobile phase, starts pushing everything along. And just like we said, the race is on. The large molecules get excluded from the pores. They just flow right through the gaps. Meanwhile, the smaller ones get temporarily sidetracked, dipping into all those pores. This difference in the path they take means the large molecules hit the finish line first, then the medium-sized ones, and finally, the smallest ones trickle out last. And just like that, everything is perfectly sorted by size. Okay, so what does this molecular racetrack actually look like in a lab? Let's take a quick look at the key components that make this whole thing work. It's actually a pretty straightforward setup when you get down to it. You've got a pump, which is basically the engine. Its job is to push that liquid carrier through the system at a very steady, constant speed. That's super important. Then you have the column. That's the sieve itself. It's really the heart of the whole operation. And finally, at the very end, you have a detector. You can think of this as the finish line, which sees and records the molecules as they come out of the column, one size group at a time. And here's a really crucial point. Not all of these molecular sieves are created equal. Scientists have this whole toolkit of different gel materials to pick from, and each one has different sized pores. So if they're trying to separate smaller things, like proteins, they might use a gel made from something called dextrin. But if they need to separate really, really big molecules, like long strands of DNA, they need a material with much bigger pores, like agarose. So choosing the right filter material is absolutely key to getting a good separation. Okay, so we've got the theory down. We know what the machine looks like. But let's get to the important part. Why does all of this sorting actually matter? Let's take a look at sec out in the real world. Yeah, this is not some obscure lab trick. Its single biggest application is purification, and that's absolutely vital for producing modern medicines. Think about drugs like insulin or sophisticated antibody therapies. SEC is a key step to make sure they are pure and safe for people to use. It's also used all the time to figure out the exact size of molecules, which is fundamental for protein research and for analyzing the polymers that make up so much of the world around us. To really see this in action, let's zoom in on one of those applications. We're going to look at analyzing polymers. And polymers are just these long, chain-like molecules that make up materials like plastics and rubber. This is just a perfect example of a real-world workflow. First, you dissolve the polymer sample. When you do that, the long chains kind of coil up into tiny spheres of different sizes. Then you inject these little coiled-up spheres into the SEC column.
And it's exactly what we saw before. The bigger spheres get excluded from the pores and rush right on through, while the smaller ones take that slower, scenic route. They come out perfectly sorted by size, which tells scientists a lot about the material. This is so important for quality control in manufacturing, making sure a plastic bottle or a car tire has exactly the properties it's supposed to have. So this ability to so neatly sort the invisible building blocks of our world, it's just an incredibly powerful tool. It lets us purify life-saving drugs, understand diseases at a molecular level, and design brand new materials. It really makes you wonder, right? Now that we've mastered this, what other invisible mysteries are we now equipped to go out and solve?